guys, this is Alicia with Good Morning Sunshine. My hair is all crazy because I forgot to brush it and I just realized it when I looked at the camera. So yeah, we're gonna deal with it for a minute. So today we had 4 lectures again and a lab. So lots of stuff again. I think we had Bacteriology 2 today, we had Epidemiology 1 and 2, and then we had Clinical Pathology 2 as well. So lots of information thrown at us, lots of different information thrown at us. So I'm going to do my best to go through it all for my understanding, not necessarily for yours because it's a lot of stuff. But yeah, it was a long day again. I have another long day tomorrow. But yeah, just barely sitting down actually. So I'm going to start with Epidemiology 1. So what is epidemiology? Well, it's the study of the distribution and determinants of health and disease in populations and the development of strategies to improve health and productivity in these populations. So there's determinants of health and disease. So the epidemiological triad, sorry, that's a big word, includes the host, the agent, and the environment. So the agents include the microparasites, the macroparasites, the toxins, and physical injury. Whereas the hosts include demographics, genetics, physiological state, immune status, and behaviors. And then, of course, environment is like climate and weather and vectors and socioeconomic and management and sanitation, so things like that. So basically, who gets sick and who does it? Where do they get sick? These are the questions we ask. When did the disease start? When did it occur? Is the population at risk? What's the location and place? So we got into epidemic curves. So basically, epidemiology is about diseases and how they spread in populations and how to keep animals healthy that are already healthy and keep them from diseases. So we look at epidemic curves. We plot frequency distribution of new cases over time, usually days or weeks. There's actually different kinds of curves. They have different patterns. So we have sporadic curves, we have endemic curves, we have epidemic curves. So that's when everyone gets exposed. We have epidemic propagating curves where the disease is still going up, which is scary, kind of like COVID in a way, because we still have cases still occurring. And then we want to flatten the curve. So basically this graph shows flatten the curve here. So we want the curve to look like in blue because that's the amount that the hospitals can take in. So summarizing epidemic curves, so type of temporal pattern, those are the types I just went over. There's an incubation period, there's a state of epidemic, and the case counts, not the rates. So how are cases connected? So connections are based on transmission. So you have direct contact, you have respiratory, you have fecal, oral, which also goes with foodborne or waterborne. We also have vector-borne, and then we have other environmental sources. So what is the magnitude of the problem? So we have types of population data. We have measurement, or also known as quantitative, which that would include like weight, hematocrit, BUN, so BUN, age, survival, time, and titter. And then we also have count, which that includes our categorical stuff and qualitative things like breed, sex, positive, negative, old, young, healthy, sick, or dead. So we got into examples. So we got into a measurement data example. So we asked, are they all normal? So are the protein values in this cattle normal? So we looked at this chart here, and this example is going to go on for a while. So it was normal for plasma and serum protein. So basically, what should I be looking at? We were looking at a green top tube, so that means we were looking at plasma protein because it had heparin in it, and that doesn't clot. It has an animal below normal range in this set of data. And then we asked for reasons for this abnormality. Well, the study was done with a refractometer, so it could have been read incorrectly, so it could be human error. These were also young beef heifers, so maybe proteins lower in these kind of cows. So also, I guess the data was taken on a really hot day, which would the cows would drink a lot of water, thus maybe messing with the data. It also looked at mean values. So here is the information basically on a graph here. So we have the mean values of 6.8 on Thursday, and then 7.0 on Tuesday. And then we have normal seroprotein, which was between 6.7 and 7.5, and then we had normal plasma protein, which was between 6 and 8. And then you can see the abnormal ones. There's one right here. So what kind of graph is this? So how many animals are sick right now? So prevalence, this would be a prevalence graph because we don't know anything about timing. So how does prevalence differ from period prevalence, from point prevalence, and then how does it differ from incidence? So incidence has to do with, So prevalence is when we don't know anything about timing, so an incidence has to do with brand new cases, like they're brand new. Sorry, I'm just looking at the PowerPoint real quick. So we have characteristics of prevalence. So it's like a photo of all diseases present at a given time. So it includes both old and new cases, like incidence just includes new cases, where prevalence includes both old and new. It is an important estimate of the scope of the problem. It's a proportion ranging from 0 to 1 or 0% to 100%. For an individual, prevalence measures the risk or probability of being a case rather than of becoming a case. So again, there's different types of prevalence. Like I was saying, there's point prevalence, which is the number of cases present at one point in time. So like slaughter check on a particular day. There's also period prevalence, which is a single examination of individuals seen over a period of time. So then period prevalence also equals point prevalence at the start plus the new cases during the period. So we have this example. So during a farm visit, you find 86 calves that are less than 4 weeks old. Of these, 8 are showing signs of diarrhea. So what is this? Is this point or period prevalence? So this would be point prevalence. So we would do the 8 divided by 86 because 8 are showing signs of diarrhea and there's 86 calves in total. So this was at one point in time. That's why it is point prevalence. So what is the rate of occurrence of new cases? So that is known as incidence, which is basically a time clock. So its characteristics are based only on animals without disease at the start of the interval, and then it includes new cases, also called incident cases. This ranges from 0 to 1 or 0 to 100% per interval. Or cases per time at risk. So it's important for predicting future impact of diseases. It measures the risk or probability of becoming a case over time. So there's 2 ways to calculate incidence. So it's new cases over animal time at risk. So you do incident risk or communicated incidence, which is new cases over the population at risk during a specific time interval. So in this example, during the last 12 months, 180 calves were born on the dairy, and 12 developed neonatal diarrhea within the first month of life. So these would be new cases because we know they're new. So they were baby cows and they got diarrhea. So these are new cases. So here, what is the incidence? We would do 12 because 12 developed the neonatal disease, and there was 180 calves, so we would do 12 divided by 180, which would give us 6.7% incidence. So how do these relate? How does prevalence relate with incidence? So prevalence equals incidence times duration. Prevalence includes effects of incidence and survival. Prevalence is most commonly used for serological studies, and incidence may be estimated by difference between sequential prevalence estimates. And then we got into some prevention. So we have primary prevention, which is the action that prevents the development of disease in an animal who is healthy. So we want to keep these animals healthy. So they're already healthy, we just want to keep them healthy. Then we have secondary prevention, which identifies animals with disease at a point early enough to prevent symptoms. So this is an animal who's already infected but hasn't shown signs of the disease yet. And then we have tertiary, which is the prevention of complications in animals who have disease. So this means the animals have the disease, but they're trying to keep them where they are. So they're trying to keep the disease from becoming worse, basically. So that was our first epidemiology lecture. I'm going to get into the second one, which is way more complicated, and she went through it really fast, so it's kind of hard to explain, so just bear with me a little bit. So for this, we are comparing disease occurrence in an outbreak. So our learning objectives here were to describe the steps in an outbreak investigation, to explain the parallels between outbreak investigation, clinical examination, and clinical reasoning. To define and calculate common measures to compare disease occurrence in populations, and interpret common descriptive and comparative measures of disease occurrence. So, expected number of cases, this changes over time and it changes over space. So what is an outbreak? An outbreak is the occurrence of disease in an area at a level exceeding the normally expected number of cases. So we have individual, which would include a physical examination, and then we have the population, which is an outbreak investigation. So outbreak or population level investigation. So 1, we want to establish that an outbreak actually exists. So this would be change in awareness, record keeping. We can see what's really going on. We can do this by observation, so new tests that come out, and then population composition, so more population is at risk. And then we want to calculate prevalence or incidence, which I just went over in the last lecture. So we have an example. Here we have these fish, which fish become more fish. So what will we use here? So this is the fish example. So we don't know when the disease occurred. So this would be prevalence. So if you didn't see, there's little red dots in some of these fish, which are counted as lesions. And we also don't know where the new fish came from. So this would also be prevalence because we don't know if they're new cases or not. So we would go with prevalence. So morbidity number 1. So that is the top fish. 1 out of 6 had lesions. So that would be 17%. And then we have morbidity number 2, so that was in the bottom group with a lot more fish. And so we saw 14 had lesions out of 20, which would equal 70%. Is this an outbreak? No, we wouldn't necessarily call this an outbreak. So then we also did the morbidity in the small pink fish in this example. So morbidity in pink fish, so all of them were sick. And also in morbidity number 2 in the bottom, the more amounts of fish, also all 14 out of 14 had lesions. So is this an outbreak? No, we would not classify it as an outbreak because it's basically the pink fish that have these lesions. So why does this matter? Well, this matters because we're exploring concurrent changes and we're evaluating economics. So we got into a sheep example. So the problem was a sheep producer has called you because he is very concerned over a sudden increase in ewe mortality in his flock. So the flock includes 131 purebred and crossbred ewes. So March 19th, there was 2 pregnant ewes, which were weak and stumbling. On March 20th to 22nd, 26 more ewes, some pregnant and some with lambs, began to stagger, walk in circles, and go down. On March 25th, 28 have become ill and 12 have died. So does a problem exist? You want to ask yourself this. And yes, a problem does exist here. Does an outbreak exist? And this would probably be a yes as well. So we would do an investigative study of this flock. Because you have more numbers, you wouldn't expect 12 ewes to have died, so in this case, so this would definitely be an outbreak. So then we got into a horse example. So you were called to the Flicka Memorial Appaloosa show. which started 2 days ago. Owners and trainers report that some horses began showing sensitivity around their muzzles, and some horses have developed blisters. So would you expect horses to have blisters? No, you wouldn't. So does a problem exist? Yes, it does. Does an outbreak exist? Yes, it does, because you would not expect this either. So, second, you would want to determine the key questions. So key questions include who gets sick and who doesn't. Where do they get sick? So location. And when do they get sick? And how are cases connected? So we ask ourselves, what do we know? What do we need to know? And how will we use this information to stop the outbreak? So we want to define the cases and non-cases. So using the same questions above, this will define non-cases and suspects. So we want to search for earlier cases and make an epidemic curve. And remember, it takes one case and then there's a pre-patent period, and then more animals will get infected if there's an outbreak. So we also want to record all events. So this is known as line listing, with line for each case or non-case and column for each characteristic of interest. So we want to include all cases and all or a sample of non-cases. So here in this example, we would have the dog's name, its gender, its reproductive status at a time of the heartworm test. We want to know if they're stray or not. We want to know their heartworm status and if they're adults or not. So we want to collect data on epidemiological factors. So cases that are normal would include a host. So that would include, again, the species, the age, the sex, the weight, the diet, the vaccination history, and occupation. The agents would include living or non-living. Environment would include sanitation, temperature, water depth, habitat, pathogen presence. And also time would include first signs, introduction to an area, exposure, partition, and death. And place would include like a pen or X and Y coordinates. Water flow, distance from woods, fan locations, and airflow. And then number 6, we want to look for patterns and associations between risk factors and disease. So these would include where, the agent, when, the host, and the environment. So example, this is a prevalence example. This is the Georgia outbreak of CIV. So here we have the factors. We have if it's adult and adult. We have the proportion of CIV positive dogs, and then we have the proportion of CIV negative dogs here. This also includes clinical signs like coughing or sneezing, how many dogs have that, how many dogs have lethargy and appetence and pyrexia. We also have if they have housemates or not, and if they have CIV positive canine housemates. We also include their vaccination history here. So prevalence or incidence. So we called this prevalence, and then we also did this kind of diagram here. So we included adults, how many had CIV, so the positive ones in adults, the positive ones in juveniles, and then we had the non-CIV in adults and the non in juveniles. So we would calculate prevalence here. So for adults, we would do 38 divided by 50, which would equal 43.2. And then for juveniles, we would do 2 divided by 2 plus 30, which would equal 6.3%. And then we would do, overall, we would do 40, and then 40 plus 80, so that'd be 120, so that would equal 33% overall are infected. So then we have prevalence ratio, and attributable prevalence, and attributable fraction exposure. I'm going to get onto those real quick before I tell you like numbers and you don't understand anything. So prevalence ratio is the prevalence with risk factor divided by the prevalence without the risk factor. And then we have the relative risk or risk ratio, which is the incidence with risk factor divided by the incidence without risk factor. So here we would just do the prevalence ratio because we're dealing with something prevalent. So then we have attributable risk, like I said, which is the incident with risk minus the incident without risk factor. Then we have the attributable fraction, which is the attributable risk divided by the incidence in a high risk group. Then we have attributable prevalence, which is the prevalence with risk factor minus the prevalence without risk factor. And then we have attributable fraction of prevalence, which equals attributable prevalence divided by the prevalence in the high risk group. So here in our example above again, so the prevalence ratio, we would then do the 43.2%, which we calculated for adults, divided by the 6.3, which we found in juveniles prevalence, which would equal 6.9 overall. So then we had the attributable prevalence, which we would also take the 43.2% we found in adults minus the 6.3% we found in juveniles, which would then equals us a 36.9 extra positive dogs. So we have 36.9 more dogs that are positive per 100 dogs. And then attributable fraction, we would do the 36.9% that we just solved for, and then we would divide it by the adults, which would equal 85.4%. So then interpretation of ratios, if it equals 1, incidence or prevalence are the same in both groups. If it is more than 1, incidence and prevalence is higher for those with the risk factor. If it is less than 1, incidence or prevalence is lower for those with the risk factor, so they're protective. So could the difference we found just be due to chance variation? So we would know this by the p-value. I remember the p-value from undergrad in math classes, and it brings back horrible memories. But anyways, so the p-value is the probability that a difference of this size or larger would be observed if there really was no difference between the groups. So again, if p equals 0.20, that means 1 out of 5 chance that it was due to chance. If p equals 0.05, it's a 1 out of 20 chance that it was due to chance. And then if it's p equals 0.01%, it is 1 out of 100 chance. So how certain are we of the size of the effect? So precision or uncertainty in estimate, confidence intervals. So here we get into confidence intervals, which is the potential range around the calculated effect size and which the real difference should be based on sample size and alpha. So here she brought in an example here. I don't know if you can see it that well. But anyways, we have the prevalence ratio here. We have, I know what that means. Hold on. Oh yeah. So it has the CI value, which is the confidence interval, and then it also has the P value. So among dogs with respiratory signs, the prevalence of CIV was about 7 times higher in adults than juveniles. So we are confident that the true increase was between about 2 and 27 times higher for adults. And this was just statistically significant because the p-value was less than 0.001. So then another step is to form the test hypothesis about disease control. So this may relate to the cause of the outbreak, the risk factors for disease, the risk factors for infection, the intervention to stop spread, quarantine, vaccination, cooling, and the treatment of affected individuals. So why test? Why not just solve the problem? This is because of cost of disease, cost of intervention, and cost of other alternatives. So then we want to plan and implement control methods. So this is based on the outbreak investigation itself that we did. So again, back to the outbreak investigation process. So we want to establish that an outbreak exists. We want to determine the key questions. We want to define the cases and non-cases. We want to record all events. We want to collect data on epidemiological factors. We want to look for patterns and associations between risk factors and disease. We also want to form and test hypotheses about disease control. And then last, we want to plan and implement control methods. So that was our epidemiology lectures for today. So I talked about bacteria yesterday in 1, and I finished the whole PowerPoint. She finished most of the PowerPoint today, and she got a little bit into what we were supposed to talk about today. So, but this one's going to be kind of short because she didn't really get into the lecture she was supposed to give today. So this has to do with bacteriology too. It has to do with host-bacteria relations. So what is a host? What is a bacteria? So environmental interaction. So there are a variety of complex relationships whereby microorganisms can interact with eukaryotic organisms known as hosts and with the environment. So a host is an organism that supports the survival and growth of microorganisms. So in veterinary terms, this is usually an animal or a bird or a reptile. but hosts can also be insects, worms, and bacteria themselves. So what is symbiosis? It is the general term for a direct association between one or more species. It basically literally means living together and therefore refers to a range of interactions including commensalism, parasitism, and saprophytism. So an example of this would be in microflora of the rumen, which were provided food and shelter while enabling the host through enzymatic processes to utilize cellulose. So what is a saprophyte? They are organisms that live on dead or decaying organic matter. Usually they're not parasites or animals, but some are capable of invasion of causing disease under the right conditions. So what is a parasite? Well, it denotes a state in which an organism lives on or within another living organism and is nutritionally dependent on them. So the parasite may or may not harm the host itself. What is commensal? Commensals are organisms that live in or on the host without causing disease. So the organism benefits from this relationship while the host may or may not. Many of the bacteria that make up an animal's normal flora are commensals. So what is pathogen? It's parasitic or saprophytic bacteria which have the potential to cause disease. These are pathogens, but they don't always have to cause disease. They just have the potential to. So it's the ability of a bacteria to damage a host is called pathogenicity. The relative capacity of a pathogen to damage a host is the degree of virulence. So what is normal flora? It's organisms including bacteria living on the host may also be called normal flora. However, some of the normal flora are potential pathogens and cause disease if there is some compromise to the host to change the host-parasite relationship. So then we also have carrier animals, which in some infections, bacteria may contribute to be shed even after resolution of clinical signs. These animals are called covalescent carriers. And then we have sites with normal flora versus sterile sites and microbes. So the sites of body that are colonized by normal flora include the skin and hollow organs whose surfaces and cavities are open to the environment. So the skin including the conjunctiva and external ear canal, mucous membranes of the gastrointestinal tract including the mouth, the oropharynx, the stomach, the small and large intestines, and the rectum. Also the upper respiratory tract to the level of the larynx, not below it. And then we have the lower genitourinary tract, which includes the distal urethra, the external cervix, and vagina. And then the bacteria, genera, and species present in these sites tends to be the host-specific and within a host or organ-specific. And all other parts of the body are considered sterile. So in healthy animals or tissues, the bacteria are present in low numbers and often rapidly cured by innate and immune defense mechanisms and do not induce inflammation. So that's all we got to in bacteriology today. We'll probably have more tomorrow because we have her 2 times tomorrow. But yeah. So then lastly, we got into animal pathology. We did descriptive gross pathology. This was actually very interesting. So our objectives for this course was to understand basic terminology in lesion description, have a general understanding of the meaning of color, distribution, shape, outline, and other factors at the moment of describing and interpreting a lesion. And also to have the knowledge to understand how and why a morphological diagnosis is made. So we started with the recognition, description, and interpretation of lesions or abnormalities. So this includes fast determination of potential problems. This is your presumptive diagnosis. And then some lesions are diagnostic or highly suggestive of a pathological process. Some patterns suggest the mechanism of disease. When lesions are not clear or there are no lesions, these findings, equally important, chart a way forward. And then we have permanent archive. These include legal investigation. So submitting samples to the laboratory. So what does this include? Well, it includes what did you see? So the gross description and exact location. What did you do? What did you think? So this includes differential diagnosis. And then what did you want? So is this neoplasm, inflammation, surgical margins, etc. So elements of macroscopic description. Today we're going over distribution, which is the arrangement of the lesion. We're going over color. We're going to outline, is it raised, flat, or depressed, and shape, is it geometrical? But later we're going to get into demarcation, which is margins, the size, so absolute versus relative, lesion, paired organs, and also texture, so the cut surface, amorphous or solid, the consistency, is it watery, soft, firm, or hard, and the extension percentage. So here we're going to start with distribution. So modifiers of distribution are focal, so that's in one spot. We have focally extensive, which this is when the lesion pushes against other organs. We have multifocal, which these are lesions in many areas. And then we have multifocal to coalescing. So this is still normal tissue in between, but many lesions on the organ coalescing because lesions are coming together. We have diffuse, which means the whole organ. We have unilateral versus bilateral. And then we have bilateral symmetrical, and then we also have miliary, disseminated, embolic, and transmural. So he just gave a lot of examples, and we had to decide what these are. So we're going to start with this one here. So this is a dog. This is the esophagus of a dog. This is a lesion in the center. So what did I tell you? So this is a focal lesion. So it's in one spot in the center. There it is. I'm not going to go over the names with you because I will butcher them. So this one's supposed to be like Spirocera lupi. I can try my best if you want to hear them, but. So next one here. This is a brain. So there is a mass here between the hemispheres. So this lesion or mass is pressing against other organs. So what is that called? What did I just tell you? So this is a focally extensive lesion. And then here, what is this? Well, this is a brain. This is a focal extensive lesion. This is on one side of the midline. So here's the lesion right here. It is also pushing against other organs. So then here, we have a chicken. This is a multifocal coalescing lesion. So as you can see, there is multiple lesions on here, but it doesn't include the whole thing. However, you can see the lesions are coming together. Some are connecting themselves with other lesions. So that is why it is multifocal coalescing. So then here, we have the goat. So this is the liver of the goat. It is not normal in color. It is accumulating fat. So this is a diffuse lesion. So diffuse means, like I said, the whole organ. So this is the whole organ. And then here we have, this is a horse. It's a horse's lungs. The left is the less affected organ, but the right is the more effective. The right is white. It's fibrin, which means severe inflammation with vascular damage. This is a unilateral lesion, so could also be said to be a diffuse unilateral. So this is the whole organ here on this side. But we have to say diffuse unilateral because it is, so if we included two, it diffuse wouldn't make sense because it's not a whole organ, if that makes sense. So here we have a dog. This is the trachea, larynx, and thyroid gland of a dog. This is a unilateral lesion. Then here, this is also a dog. This is the thyroid gland with the parathyroid gland. This is a bilateral diffuse lesion of the parathyroid gland because the whole parathyroid gland is infected by the lesion. So then we have, this is a dog. This is the gastric mucosa. This is multifocal. If it goes through the wall, it is known as transmural lesion. So it has to go all the way through though. So then we got into color. So we have white, which means inflammation, necrosis, mineral, keratin, fibrosis, urates, lipids, and bone. Then we have the color red, which includes hemorrhages, congestion, and hyperemia. And then we have yellow, which includes inflammation, bilirubin, fat, myelin decomposition, mechanomium, and fibrin. And then we have green in color, which indicates biliary, pigments, eosinophilic, inflammation, necrosis, allergy or fungi, or hemosiderin. And then we have the color black, which includes melanin, blood, necrosis, fascicula, pigment, exogenous, and carbon. So again, we're going over examples again. So what is this? Well, this is a dog. This is the dog's kidney. This is a multifocal coalescing lesion. The color is red. These are really small, so it is round to regular lesions. But yeah. So here in the horse, this is a horse's intestine. There are actually 2 lesions. So we have the lesion right here. We also have the lesion here. So we have an abnormal intestine. The color is red. A segment is abnormal, not the whole thing of the intestine. Then there is a fat lesion. This is the lipoma. The lipoma, it introduced an entrapment of the intestine. So this would be an emergency surgery. Blood enters but doesn't come out. So this is known as congestion as well. Here we have a calf. This is the trachea of a calf. The color is brown and green. This is fibrin, which means inflammation. And this is diffuse fibrinal tracheitis, because it includes the whole organ. And then we have an alpaca here. This is green, yellow in color. Its distribution is multifocal to coalescing in the mucosa of the esophagus. There are little holes that don't go all the way through, so they are ulcers. And then here we have a dog with icterus. This is yellow, but bilirubin is accumulated and prehepatic. It's either prehepatic, hepatic, or posthepatic that is causing this. Here we have a dog. We have a sonoma or a Leydig cell tumor. This is a testicle of a dog. It has 2 masses actually. So we have the big white mass right here, and then we also have the other yellow mass right there. And the moon shape right here is the normal part right here. So 80% is the white focal nodule. The yellow nodule, which produces hormones, is well demarcated. So here we have a cow heart. It has induced eosinophilic myocarditis. So the first image is a heart, like I said. This is a multifocal to coalescing here. And here he found a rotisserie chicken at Sam's Club, I guess. This is green. It is a focal lesion here. It's deep in the breast muscle. It's deep. This is known as deep pectoral myositis. This is due to big birds flapping their wings, which produces ischemia in the breast muscle. So then we got into outline. Is it raised or widened? Something was added. Be fluid, which means edema or hemorrhages. Have cells, which means inflammation, neoplasia, and normal elements. And then we have gas, which is usually bacterial. So again, more examples here. We have a turkey. This is known as avian pox. It is multifocal to coalescing. This is well demarcated and it has raised nodule. This is also known as dry pox, not the wet pox. So here we have a dog. We have a melanoma. This is the lung of a dog. This is a multifocal to coalescing, as you can see here. This is black. It is slightly raised and well demarcated nodules. And then we have depressed. So something was removed. So we have like necrosis, hypoplasia, aplasia, and atrophy. So in the cat example here, this is feline ischemic encephalopathy. This is a brain. This is unilateral. This is atrophy. This is a unilateral atrophy of the cerebral cortex in this cat. So here we have a pig. This is the liver of a pig. The liver has depressions. I don't know if you can see them, but it's multifocal to coalescing wide depressions. So then we got into shape. We started on shape anyways. So shapes can be triangular, rhomboidal, spherical, amorphous, cavitated, and linear. Triangular usually has a vascular origin or tip of the triangle at the occluded vessel. So here we have a dog. It has acute renal infarct. This is a kidney. This is the dark red black in the cortex. The center is in one of the vessels, like it said. So rhomboidal, also vascular in origin. So the pig has cutaneous infarcts. Then we got into spherical, vascular cyst. He didn't go over these, so I haven't learned these ones yet. But I think he said he was going to post the rest of the lecture because he missed a few. So I'm not going to go over with you because I don't really understand it yet. But anyways, those were our lectures for today. And then we had lab. Lab, we learned how to do blood smears, but I already knew how to do those because of work. And then we also did PCVs, which I also knew how to do, and how to read PCVs and total solids. And then we were looking at gram stains. And then I think that was it. But yeah, thank you guys for listening. I know it's a lot of boring material. Imagine sitting through that. But yeah, another long day tomorrow. So, but I hope you guys had a good day today. I still have a lot of work to do, like always. But yeah, I will talk to you guys tomorrow about new lecture material.